The overall goal of the following experiment is to screen genetically and pharmacologically manipulated mice for malfunctions in basic mechanisms of cognition. This is achieved by making psychophysical measurements of physiological meaningful quantitative properties in a fully automated high-throughput 24/7 behavioral phenotyping system. The next step is to analyze and graph the incoming data in quasi-real time, which enables automated real-time control of testing on a mouse-by-mouse basis. Next, the raw data, the test protocol code, and all intermediate statistics are archived in a single data structure in order to make the data publicly available and encourage further analysis. The results show strain-specific alterations in the temporal Weber fraction based on the switch task.
The main advantage of this technique over other methods such as the water maze is that it gives you physiologically meaningful results, very high throughput, and no handling of the mice.
So this method can help answer key questions in the cognitive neurogenetics field, such as are there mutant strains that have quantitatively altered interval timing mechanisms.
The implications of this technique extend toward therapy or diagnosis of genetic cognitive deficits because it may lead to the discovery of genes that specify critical components of these mechanisms.
Individuals new to this method need to understand the basics of MATLAB programming and to understand the power of quantitative automated psychophysical testing.
Adam and I first had the idea for creating this system when we saw the labor-intensive and unquantitative nature of the